Hello everyone. Uh, we are in module 6 now, and this is the second lecture of module 6. Here I am going to discuss about pattern formation and growth. During the last lecture, mostly I was discussing about how different types of cells are established in the body of multicellular organism. Different types of cells, how they are getting originated. Today, whatever I am going to discuss, this is little bit more complex. Now, if you see, I already mentioned during my first lecture in module 1, there I mentioned that although the basic or the fundamental understanding about different types of cellular processes like transcription, translation, those are very much conserved in different types of organism, including plant, animal, everywhere. But if we go little bit, I would say upward. Upward means here, in the level of cell, tissues, and then as a whole, the whole whole animal or plant body, you will see different types of variation there. So here, whatever I will explain now, you will see lot of variation even in a phylum. For example, how like I would say bird, how amphibian or mammals they get developed, how their pattern are actually formed. So those are kind of huge variation there. Let's see. So mostly I will be discussing about these 3 points here today, understanding body plan and complex organization, followed by the molecular basis of pattern formation during early development, and finally understanding growth of an organism. So here, if you see that what I already mentioned, that different animals use different mechanisms to establish their primary axis of polarization. So this is, the major idea here is, if you see, like when animals they are getting developed, 3 major axes should be should be created in proper timing. So the first one is, I would say, number 1 is animal vegetal axis. So 3 axes I told, this is one animal vegetal axis. The second one is dorsoventral axis, and number 3 should be anterior posterior axis. Anterior posterior axis. Now, if you see those 3 different type of axes, so the problem here with the naming is animal vegetal axis. There is nothing to do with vegetable or anything. This is some kind of weird name. That's why all these things actually determine, I would say, animal vegetal axis means during development which portion will be internal and which portion should be external. This is just animal and vegetal axis. And dorsoventral, you understand from the name itself. So that is dorsal, that is the back side, and ventral, the front side. For example, in human, if you compare, and the front side is the ventral part and the dorsal is the back side. And the third one is anterior posterior axis, so head and tail part. So that's all. These 3 major axes should be established during the development, I would say during the early stage of development itself. So now here, if you see, this is the side view of a Xenophus egg. So now like particularly this egg cell, the one important thing is, if you see egg from face, frog is an example of amphibia. So frog or birds, their egg size, they are kind of much bigger. But if you see the egg of mammals, particularly egg cell of human, it's a very small compared to egg of this fish, amphibia or bird. Now, if you see, there are a lot of information already present in egg which dictates the future developmental plan. So here I am just showing, so I will not go through all the details here, just the basic outline, basic features I will discuss. So here if you see, this one is an egg of xenopus. So this is kind of frog egg. And even before fertilization, as you can see, this is the sperm here. So sperm here. But even before fertilization, you can see some kind of asymmetric distribution of molecules inside the egg. So as you can see here, I am mentioning here that VEG-T mRNA, and then here went 11 mRNA. So there are a lot of stuff. So those two are very well characterized. So we are mentioning here that some kind of asymmetric distribution. As you can see, this same mRNA is not present in this side, but it is present here on the vegetal pole, but not in the animal pole. So now what I am trying to explain here, this information is already present in the egg itself. The zygote is not yet formed, the fertilization not yet happened, but still those informations are properly organized. And now this is the entry point of the sperm, and after sperm entry you can see there is little bit of rotation in the egg cortex. So as you can see here, so this is egg cortex, this is the egg cortex layer here. So here you can see some kind of rotation in the egg cortex. So that will lead to further changes to the asymmetric distribution of molecules to define a dorsoventral as well as animal and vegetal asymmetry. So as you can see, now we are also mentioning, in addition to animal and vegetal pole, now we are kind of mentioning dorsal and ventral side. So still it is a single cell, but when it will divide into multiple cells and it's going on, by that time those things are already decided, the dorsoventral axis as well as animal and vegetal axis. So this is kind of whole idea about this this slide. The next thing here, so I was discussing about frog egg, but here in this slide I am trying to explain something else with Drosophila system, with Drosophila egg. So at the beginning I mentioned that you will get several types of diversity in this developmental pathway, like whether you were taking some pathway from frog or Drosophila or human, there will be many changes there. So here if you see studies in Drosophila particularly, it is a model organism and it revealed the genetic control mechanism underlying development. So here this is the fertilized egg of Drosophila. So Drosophila egg is not kind of very much spherical, it is kind of oblongate shape. So as you can see here, this is fertilized egg. Now the beauty here is, just after fertilization this cell will first not divide. Actually the nucleus, that it will divide very rapidly and we will get a cloud of nuclei. So as you can see, there are many nucleus inside this zygote, I would say. It's already fertilized egg. So you will get cloud of nuclei inside. And then over the time, this nuclei will migrate to periphery, and where the cell boundaries will eventually form. So in this figure, as you can see, cells are present in the boundary. So these are cells. So just the cell membranes are getting generated here. Not yet fully formed, but it is starting to be generated now. But now when the number of nucleus is about 6,000, the plasma membrane grow inward to enclose its nucleus, resulting in I would say approximately 6,000 cells. Although in this diagram, for example here, you will see this is just in 2 dimensions. That's why you can see those cells are present in the boundary. But if you compare in a 3-dimensional shape, in all the periphery, they are actually now covered by a layer of cells, by these cells, whatever I just mentioned. And here, another important thing, at the one end, you will get approximately 15 nuclei in the posterior end, and they will finally form the germline precursor, the germ cells they will make in future. So this is the beginning of Drosophila embryo development. So at the beginning, how it is getting developed. So here I just mentioned how initial nuclear division followed by membrane formation. It's overall how it developed from an egg to embryo. But now we have to understand how this axis being formed. Whatever I mentioned at the beginning, that for example anterior and posterior axis, how head and tail will be defined. So let's see. Some egg polarity genes encode macromolecules deposited in the egg organize the axis of early Drosophila embryo. So I will explain this. So again, this is, I would say, just a single cell. You can consider this is bicoid mRNA is deposited at the anterior pole during oogenesis. That means this egg is not yet fertilized. This is even before fertilization. Okay, even before fertilization, you can see at one end here. So something is getting deposited. So lot of research on this topic that actually establish that this is whatever is getting deposited, that is some mRNA. It's called bicoid mRNA. So this is bicoid mRNA. Somehow this bicoid mRNA is restricted to one pole. So if this mRNA is restricted to that pole, so mRNA is getting translated. So therefore, we will get some kind of concentration gradient of protein. So here, because the mRNA is present here, so there we will get more kind of translation there. And after translation, those proteins, that bicoid protein, they will be slowly diffused through the cell inside the egg. So as a result of that, you can see here these are bicoid proteins, molecule. But this local translation followed by diffusion generates the gradient of bicoid protein. So as a result of that, here you have a very less amount of bicoid protein. Here I would say moderate amount, and here very high amount of bicoid protein present. So it will make some gradient, and that will actually dictate the anterior and posterior axis in Drosophila. Particularly bicoid proteins or bicoid mRNA, whatever they are concentrated in one pole, that should be the head of Drosophila. So this is finally this end will be the anterior side, or it will finally this side will be a head part after so many rounds of cell division is going on and on after that. Now in this curve, we are trying to explain you that this is the anterior side and this is the posterior side. And now if you see the bicoid protein concentration, as I already mentioned, so this blue color curve here, this is the bicoid protein concentration at the anterior end. That means this side you have more bicoid protein. So you can see that gradually when we are going towards the posterior side, the amount of bicoid protein is getting low. But the red curve here, as you can see, there is no changes, because this Drosophila is a bicoid mutant. That means if the bicoid mutant means they are not making the bicoid protein. So as a result of that, the anterior side will not be decided. So this is just for study this developmental pathway. Scientists, they generated this mutant. So now I just mentioned about this bicoid, and this protein is getting deposited, I would say, making some kind of gradient, and that makes the anterior side of Drosophila. But now similarly in the posterior system, in the posterior side or in back side, you will see another mRNA which is getting concentrated. This is called nanos. This is called nanos, and it will make some kind of nanos protein also, and exactly just like bicoid, but this is opposite, that this protein will be more on the posterior side, and it will be gradually, it will go low and low towards the anterior side. So as a result of that, if I try to understand it a little bit in more detail, so here I am just mentioning in x-axis. So here on the left side, this is the anterior side, and on the right side, this is the posterior side. And in this y-axis, if we are showing here that bicoid protein concentration, and in this right hand y-axis, nanos protein concentration, then how it looks like. So it is very simple, as I already explained. So in case of bicoid protein concentration, I would say it will be going something like this in normal Drosophila. No mutant, nothing. This is the concentration of bicoid protein. Similarly, nanos protein will be something like this, just opposite here. I am just mentioning in dotted line for understanding purpose, something like this. So this will be bicoid, and this is nanos concentration. So there are lot of things there. Many other things are also involved. I just wanted to explain few of them so that you will get some kind of flavor that how those things are happening, how these complex processes are happening during developmental processes. Now, we just mentioned at the beginning about frog embryo, like we started with egg of some amphibian egg, and then next we discuss about Drosophila embryo, and then here we are going into some example with I would say something like mammal. So early developmental events in mouse embryo. This is, if you see the whole slide, it is very complex. I will only mention some major point in context to whatever I have already discussed. So as you can see, in case of frog embryo, particularly I would say frog egg, as well as in case of Drosophila egg, already it has been determined some of their axis because of their that asymmetric distribution of substances like mRNA as well as proteins. But here if you see early developmental events, so here at the very top, whatever we are showing here, that E 0.5, E2, E2.5, this is nothing but embryonic age, right? Embryonic age. And then there at the beginning, we are showing that this is zygote. That means one thing we have to understand, whatever I mentioned about frog egg as well as Drosophila egg. So in that case, the female frog or female Drosophila, they just lay egg outside externally. But in case of mammal, everything happen inside the body. So as a result of that, after fertilization of the egg, so that egg should be develop into embryo, and it will be implanted inside the uterus. So there are huge difference between frog, Drosophila, and some example from mammal, for example mouse, human. So as you can see here, the zygote. So zygote means still one cell, but already fertilized. the egg and sperm already, the fertilization already happened. So here at the very, here in the lower panel, we are trying to show you that morphogenetic events, what is happening here. So this is the zygote, and how many cells present there? Just one cell. Just fertilization happened, and then it is going like this. Some cell division happened, then 4 cells, then 8 to 16 cells. It's going like this. But remember, in case of Drosophila, I mentioned that initially just the nucleus divide. But in this case, no. Normal cell division is happening. So as a result of that, you can see, I would say at around 8 to 16 cell stage here, which is called morula. So in this stage, all cells, they have the potential to make everything. In case of Drosophila egg, I mentioned that even before fertilization, that bicoid and nanos concentration dictates that which side will be the head and which side will be the tail. But in this case, as you can see, at this level, morula, so 8 to 16 cell stage, but that time also everything is very much similar. They have the whole potential here. Now, if we go further forward towards developmental side, so then you will find the blastocyst. So now from this point you can see here some inner cell mass, that ICM, inner cell mass, is getting generated, and then slowly you will see lot of stuff will be getting established. For example, since I mentioned about the anterior and posterior pattern, so you can see here at this stage when we have around 120 to 250 cells in this stage. So their anterior posterior patterning is happening in case of mouse. So this is the difference between Drosophila embryo development and here in case of mouse. And slowly it will go towards the developmental, further developmental stages, and you can see at when the cell numbers are very high here, so almost 60,000 to 90,000, from that point of time the organogenesis will start. So as you can see here, the noodle fold is being prepared. So that way it is going on. So this is kind of, I would say, some kind of like very brief idea about the differences between the embryo or the patterning formation in case of frog, Drosophila, as well as in mouse. Now, whatever I mentioned, that at the beginning 8 to 16 cell stage, they have potential to make anything. But in case of Drosophila, that is not possible, because already those are defined in egg itself before the fertilization. So now, in order to corroborate whatever I explained in the last slide, so I will give you some like example here, some real life example. For example, the monozygotic twins explains the developmental potential of cells during very early embryonic stage. So for example, in human population, you see this monozygotic twins. That means I would say there can be 2 different types of twin, whatever we see in our natural population. One is monozygotic twin, so that means they are identical twin. Both of them, they are originated from same zygote, that is monozygotic twin. So I'm just writing here, this is identical twin. And the other one is dizygotic twin. So that is something else. During the same time, actually, 2 egg cells get matured and both of them fertilized by 2 different sperm. So as a result of that, those 2 zygote are different from the beginning, and that will lead into non-identical twin, right? Non-identical twin. So that is nothing but 2 different zygote form at the same time, and both of them they get matured, and that is they are forming 2 babies. That's all. But now, how this monozygotic twin actually develop. That means after fertilization, when the zygote form, and this zygote when they are getting divided, I would say when they are very early stage, around 8 to 16 cell stage, during that time accidentally it can happen naturally also. So if that like early embryo gets splitted and they are not yet implanted on inside the uterus, so as a result of that, when this splitted embryo, which will which will be implanted inside uterus, they will develop two like identical twin. So similarly, the same idea, whatever I told, this is kind of natural observation, but if you see that has been experimentally demonstrated also, documented also. So what people did, so they scientists, they use SIP and they took egg cells from SIP and then it's undergone IVF, in vitro fertilization, and then embryo is getting formed inside lab, right, in vitro system. So when this is just 8 to 16 cell stage, during this time, surgically, if they like split this embryo into two, so this is splitted embryo, but this is surgically conducted, right? This is splitted embryo. And now, if this embryo are implanted in surrogate mother, in this case SIP, then it will give rise to birth, like it will give rise to I would say identical SIP, just like our monozygotic twin. So that suggests that at the very beginning of this mammalian developmental stage, when like only few cells are present, they have full potential to develop everything. That's why they are being normal, like after even the splitting, that anterior posterior system, dorsal ventral system, everything is still like going normally. So that's kind of a huge difference between frog, Drosophila and mammalian system and their developmental processes. Now, in this part, I will be discussing mostly about growth. So, how growth is controlled, right? So, this is very interesting. As you can see, sometime like if I compare human with elephant, so obviously elephant is huge compared to human being. So elephant might have so many cells also, but sometimes we see even in same species there is drastic difference. For example, here this dog, some of you may know that this is Great Dane. It's a huge dog, Great Dane. And here you can see here a small dog present here also in black color, as you can see. So this is Chihuahua. So this is just very small dog actually. People like it for their size. And both are dog. They are same species, but as you can see, they have massive difference in terms of their size. I would say almost 35 to 40 times difference in terms of their weight, this Chihuahua and Great Dane. Chihuahua is approximately 3 to 4 kg weight, right? It's very small dog. Now, here, if you see, like there are multiple variables that define the size of an organ or organism. The size of cells, that is one, whether cells are small or bigger, and the number of cells and the quantity of extracellular material per cell. So as you can see, so here and here, So number of cells same, but somehow if cells they grow in size, then the overall volume is increasing here. So this is also kind of cell growth. And now cell division in the second case here. As you can see, here you have total how many? Total you have 7 cells present. Now out of those 7 cells, 4 of them here you can see they are undergoing division. So as a result of that, 4 plus 4 8 and 8 plus 3. So total now you have here 11 cells. It is also increasing, but this is the number of cell increasing because of the cell division. And differences in size means it can grow, or sometime because of some reason some cells may die also, right? So that is cell death. So we have some mechanism called cell death mechanism, programmed cell death or apoptosis. Because of that you can see here also we have here 7 cells and 3 of them undergoing apoptosis or cell death. So as a result of that, now you have 4 cells present here. And finally matrix deposition. That means here the number of cells and here number of cells same, but somehow in between cells, the space in between cells that is growing because of matrix deposition. That will also increase the overall increase in volume, in size. Okay. So now in order to understand more about this growth of an organism or of an organ, like scientists, they carried out beautiful experiment. So here I am going to explain one of those experiments. So the transplantation experiments provide insights into the growth of different organs. So you can see here the fetal thymus. So thymus gland is I would say part of our immune system. This is some primary lymphoid organ present just our upper part of our chest, just behind our sternum, the main bone here, and just on the close to heart actually there present one organ, it's called thymus. Here T cells they get matured. So I will discuss that in detail during the immunology discussion. So these thymus glands, when transplanted into developing mouse, what happened? Multiple thymus gland actually transplanted into some developing mouse, and each of those grows to its characteristic adult size. That means when they transferred, they saw that these thymus are growing into normal size. The multiple thymus is getting developed, right? On the other hand, another organ, this is also part of our immune system, the spleen. This is secondary lymphoid organ, right? Here many immune cells they get stored in our body, so inside our body. So now multiple fetal spleens are transplanted into a developing mouse. Same experiment, but instead of thymus, here we are transplanting spleens, but the result is completely different. As you can see, each of those they end up smaller than normal spleen size, but collectively they grow to the size of one adult spleen. So this is very interesting that all those spleen they didn't grow, like they are very miniature size, but if you add up all those spleen together, you will find their total mass, total size should be a single spleen of an control mouse. So as a result of that, what we would like to conclude that the thymus growth is regulated by local mechanism intrinsic to the individual organ. So as a result of that, although we have transplanted multiple thymus glands, but all of them they grow in normal size. But whereas the spleen growth is controlled by a feedback mechanism that that senses the quantity of spleen tissue in the body as a whole. So as a result of that, you can understand it's a very complex process that in the same individual, in case of mouse in this case, the spleen growth and the thymus growth, they are controlled by different ways. And now I would like to give you some other example also. So another example here, this is C. elegans. So this is again a model organism, C. elegans. This is a worm. So now in C. elegans, like this is like very interesting because this is a model organism for studying developmental biology that follows and like astonishingly precise and predictable developmental program. So this is like, we know a lot about C. elegans, and this is also known that in C. elegans we have 959 somatic cells. 959 somatic cells. It's so precise, so fixed. The number is so fixed. 959 somatic cells. That means not the germ cells are included here, just the body cells. But based on the cell division, because it's a model organism and scientists know a lot about C. elegans, but based on the number of cell division, the actual numbers of somatic cells should be 1090, 190 somatic cells should be there. But what I am telling now, so 959 cells present, because 131 cells, they get deleted by the method called apoptosis. I will discuss this in more details in some other classes. So as a result of that, now if you add up 959 and 131, then you will get this 1090 somatic cells in C. elegans. But the point here is, like when this nematode, these C. elegans, they attain sexual maturity, the number of germ cells, that might be different, but their somatic cell number is fixed, 959 cells, when they attain sexual maturity. After that, I would say like 1 or 2 weeks later, if you see the size of the C. elegans, size of this worm get doubled, but the number of somatic cells remains same. Then what is the reason? The number of somatic cells remains same, but the size of this organism, that is getting doubled. That is because the like these cells, only 959 cells, but they grow in size. As I explained, there are multiple mechanisms. In this case, cells, they are growing in size. But why? Why they are growing in size? Because some some more complicacy there. The like one explanation here is that although the cell is not getting divided, although they just have 959 cells, but the nuclear division, the DNA synthesis is happening, not the nuclear division, I would say the DNA synthesis is happening. So as a result of that, those cells are now polyploid. Ploidy number means the number of genomes present per cell. So ploidy number increase, the chromosome number increase in each of these somatic cells. So as a result of that, those cells are getting huge. They are making more and more product, but the number of cells remain fixed. So this is another way how cells can grow in size, and as a result of that, the whole organism here, in this case nematode, this C. elegans, it is getting doubled in size, although they are maintaining their I would say their total number of somatic cells. This is the last slide. Here I would like to mention that the proliferation, death and size of cells determine the organism size. So I just mentioned in the previous slide that how size of cells actually help to nematode grow. In this case, this idea, this increasing ploidy number and in terms of, I would say, their size is growing. That is very important in context to our, I would say, in context to our agriculture, because many of the vegetables we are nowadays eating, like many of them, they have increased ploidy number just to make bigger vegetables and more productive, that's all. So as you can see here, this is and I would say this is a plant. This plant is Arabidopsis. This is again a model organism, Arabidopsis. So today I was discussing about frog, then Drosophila, then mouse, human. There are so many organisms and their different features. So Arabidopsis, for example. So now in this panel, as you can see here, the cells are much smaller, but here cells are much bigger, because here the ploidy number is more. The genome number per cell is more there. So as a result of, cells are getting bigger. But finally, what will happen because of that, if you see, so and these cells, whatever I am showing here, so those cells are from petal. Petal means the parts from their flower, right? So this is the petal. So as a result of that, now you see the flower size. This is the normal Arabidopsis plant. Their flower size is much smaller, and as a result of that, when this ploidy number is more, cells are much bigger. You can see here this flowers, Arabidopsis flower, this is much bigger compared to the normal Arabidopsis flower. But this is in case of Arabidopsis. But now if I give you another example, you will be seeing something different. So here, whatever this tube, hollow tube you can see, this is part of the, this is part of their kidney tubule in case of salamander. This is salamander. Again, this is an amphibian, just like frog. So this is salamander. If you see their kidney tubule, so here in this tubule, you will see multiple cells are present. And here the cell numbers are going low, here only few cells present, but still they are forming the tube. But why this is happening? Because as you can see in this case, if you see, so if you see the gross anatomy, the whole body of this salamander during the early stages, you will be seeing their overall size, volume, they are same. But if you see their renal tube, that is also same in terms of their size, in terms of their volume. But the number of cells getting low when the ploidy number is more. As you can see, this is haploid, this is diploid, and this is pentaploid. So whatever we observe in case of this plant Arabidopsis, this is not happening here in case of salamander. Here, this increasing size is getting compensated by lowering the number of cells, so that they can control the overall volume of this, I would say, salamander, overall size of this salamander, and everything is happening properly. So as you can see, there are a lot of detailed mechanism about this growth control mechanism. I just mentioned few of them, few interesting aspects of them. And if you want to understand in more details, you can follow any good developmental biology textbook, or for example this book, particularly I am referring in this case, The Molecular Biology of Cell by Alberts. That's all.